You sitting here?
Get up soon, right? Welcome back. Welcome back. Yeah. Okay. Is it working?
Yes, working.
All right. Great. So there's been a bit of a change in the schedule. We have Chandrima speaking instead of Nishana, and Nishana will speak later in the day. Right. So Chandrima Das is from Saha Institute, and she's going to talk to us not about nuclear physics, right? No. Okay. All right. She's going to talk about that's Saha Institute of Nuclear Physics. Anyway, so she's going to talk about chromatin readers.
Hello. Okay. Thank you, the organizers, particularly Dimple, Ranjith, and Mithun, for inviting me here. And it's a very pleasant kind of occasion to share our work. It's a beautiful campus. I have been longing to come here for a while, and it's really good to share our work experience. So I am from Saha Institute of Nuclear Physics, Kolkata, and we are working on epigenetic regulators with a class of proteins called chromatin readers. So in the complex genomic organization, the information needs to be decoded, and there are multiple factors that lead to epigenomic regulations. To make a very simple representation, these histone tails, which has these modifications, have the writers which puts the marks, erasers which removes the marks, and we are working on the chromatin readers which are involved in this kind of recognition of these modifications. So very simply, what are chromatin readers? These are kind of a barcode readers, and they can actually understand and kind of regulate these epigenomic modifications. So in the nucleosomal context, these post-translational modification, acetylation, methylation, phosphorylation, even proteins like ubiquitin moieties are recognized and kind of translated by these chromatin reader class of proteins. So basically in our work, we are focusing on human disease and mostly in the cancer context. And we have in the epigenetic toolkit a class of family of proteins like Zemin date, UBR7, transcription factor 19, speckle 110. These are some of the candidates, and we are looking in the different cancer context. For today's talk, I'll be focusing on the breast cancer aspect. And what are the kind of questions we are looking at? How the gene expression regulations are being fine-tuned by these readers in cancer cells. We are also looking at some of the other different problems, like how the cancer cell's extracellular matrix is contributing to the tumor cell niche, and also the invasive and metastatic potential of the tumors, and also metabolic reprogramming, and how these epigenetic regulators play a seminal role in that. So I'll take you to the first part of the talk. The first is the metabolic regulation and the role of the readers. So this is a tumor kind of three-dimensional representation, and it is needless to say that the chromatin domains in the core and the core of the tumor versus the different regions, the peripheral cells, are very different. So there are different metabolic programs that are actually reinstated in the core of the tumor versus the periphery of the tumor. So as you can see, the core of the tumor is an oxygen depleted, nutrient depleted center. It's a hypoxic environment, and the glucose is basically converted to pyruvate, and then there's an axis which shifts it more towards lactate. So we are actually working on this kind of metabolic shift that is happening in the hypoxic tumor core environment, which is contributing to the proliferative potential, chemotherapy resistance, and other properties of the cancer cells. So we are working on this factor Zemin date for the last couple of years, and there are different school of thoughts which kind of projects this zinc finger type of protein. So there are roles and models that are established it as a tumor suppressor protein. So it could be a kind of modulus, kind of ATRA-mediated regulation that has been well studied, and also other interacting partner switch mechanisms are at place. It shows that it binds to KDM family proteins, and in different contexts it can promote, it can inhibit proliferation, oncogenesis, and the metastatic potential. On the other hand, there are groups which have started working on the tumor promotion models, and there is a paper that shows that in the hypoxic environment, the hypoxia response element sites, which are recognized by HIF1 alpha. This recruits the D8 containing complex and it promotes angiogenesis cell motility. So we are asking the question, moving from 2D to 3D context, how this ZMIND8 is involved in tumorigenicity regulation. So we started our experiments very simply from the clinics. We dissected the whole tumor models in the core region and the peripheral region, which are different molecularly, and this is the surgeons who can do this dissection and clinically give us these samples. And now when we looked into this tumor periphery versus core, we were surprised to see the ZMIND8 is one of the factors which is quite heightened in the tumor core. So indicates that particular microenvironment that is there is being fine-tuned by this ZMIND8 protein. So we moved ahead and established the 3D spheroid models in the lab, and you can see in this spheroid context, we first used the molecular markers like Ki67 and CA9 to establish the periphery and the core of the tumor. And then if you look at the ZMIND8 expression, it is again mostly confined in the tumor core region. So molecularly, something is going on with this epigenetic regulator in the tumor cores. So now we started mimicking this environment of the kind of core-like conditions by two ways. One is initially we started with low glucose and cobalt chloride treatment to activate the HIF1 alpha, and then later on we moved to this hypoxia chamber-mediated hypoxia activation, and you can see in HIF1 alpha activation quite robustly happening. What happens to ZM/ND8? So you can again see in both the conditions there is aberrant, there's a significant overexpression of ZMIND8 protein, so indicating it's a hypoxia sensor. So we did RNA sequencing analysis, knocking down ZMIND8, and out of the several biological pathways that has come up, one of the areas that we have focused on is the metabolic regulation aspect. So, so then we went ahead and performed chip sequencing from the core cells of the tumor, and you can see here they have the general recruitment in the promoter TSS, and again metabolic processes came up as major biological pathways. And again, very interestingly, when we looked at the kind of pathways through gene set enrichment analysis, we find a lot of genes are involved in the glucose metabolism regulation, including the hypoxia-like conditions that are involved here, glycolysis, gluconeogenesis pathways, and several other factors that kind of corroborates with the glucose metabolic changes that is happening in the tumor core. So we went ahead and performed immunohistochemistry analysis from these different tumors, and you can see the ZM/ND8 and HIF1 alpha has a beautiful correlation in these tumors in the context of expression. We also saw that in the tumor core, ZM/ND8 and HIF1 alpha does physically interact, and there are many target enzymes that ZM/ND8 regulates. Like in hypoxia condition, you see ZM/ND8 being elevated along with HIF1 alpha, and it leads to activation of LDHA and hexokinase 2. We again connected with our ChIP-seq data, and you can see HIF1 alpha and D8 being co-recruited in these important metabolic enzymes like LDHA, HK2 genes. So indicates that it's a co-recruitment model where both of these factors get recruited together and is regulating the glucose flux. So getting into the glucose flux, the first thing we did is looked into the extracellular acidification rate in the core tumor cells where we knocked down D8, and you can see there is a downregulation of the ECAR as kind of expected. And we also saw that if you target ZM/ND8 in these cells, the LDHA, which is the master regulator to pyruvate to lactate transfer, this expression is highly downregulated. So this, along with this, we also measured the lactate pools in the cells. So these are the lactate pools, which shows again significant reduction when you target D8. And I haven't shown here, but acetyl-CoA levels show the reciprocal, that is where the core condition you target D8, these lactate levels are downregulated, acetyl-CoA levels get upregulated. So we finally went ahead and performed the PBMC invasion assays, where we take the PBMC cells and from the different conditions, the norpoxia where D8 is knocked down versus hypoxia again where the knockdown is done, we add these kind of cell media and look at the effect of the PBMC invasion. And you can see here, this is very specific to the hypoxia context, where in the hypoxia you have a low level of invasion, but when you knock down this ZM/ND8, there's a heightened invasion. So bringing the things into a simplistic model, which we are doing, and so you can see that glucose to pyruvate and pyruvate to lactate transition in this Z-MINT8 is playing a seminal role and diverting the metabolic flux towards the lactate. There is more lactate release, and then that can kind of suppresses the immune cell calling, and you can see that this is really a factor in the tumorigenic context. It is an oncogene in the context of the hypoxic tumor environment. Okay, so this is the part one of the story, and then I would take you to the second aspect, which I have mentioned, is extracellular matrix remodeling and how the factors epigenetic readers play an important role. So this is a quick introduction of the normal extracellular matrix and stiffened matrix in cancer. So what you see is there are lots of changes that happens in the matrix level, like collagen bundling and cross-linking and other things that really lead to stiffness of these extracellular matrix in the cancer. So these stiffened matrices have multitudinal functions that it kind of evades the growth of tumor suppressors, resists cell death, escapes immune response, acts as a drug-deficient barrier, and induces angiogenesis. For today's talk, I'll be focusing on this promotion of the invasion metastasis, although we are doing the respective other functions and the role of the ECM in this context as well. So this is kind of the ECM deposition and ECM alterations that happens. So the first phase in the cancer cells is ECM deposition, followed by post-translational modification leading to ECM cross-linking. That is very important. And finally, the ECM degradation, which is connected with this angiogenesis phenomena. And if you look at the cancer, it's kind of, we kind of speculate that we could actually bookmark the cancer, whether it's in carcinoma in situ, the basement membrane breakage, and invasive tumor context with this ECM stiffness and rigidity and the context of the ECMs that we are studying. So we have been working with one important protein called E3 ubiquitin ligase UBR7, and we initially had showed that this UBR7, although it's a chromatin reader family, has acquired different function. It's an atypical PhD finger containing protein. It acts now as a H2B lysine 120 ubiquitinating enzyme. And we found that if you target the UBR7 in the cells, lysine 120 ubiquitination dramatically drops. And this has direct consequence in the cellular invasiveness, alteration in the EMT programs in the cells. With the ECAD, the epithelial markers gets down-regulated, whereas NCAD or Vimin10 gets turned on. And in the animal model experiments, you can see that UBR7 wild type overexpressed tumors are kind of, it is a suppressor, and the tumors do not come up so easily. We also looked at the patient level, the primary versus metastatic tumors, and see that there is a downregulation of UBR7 expression in the metastatic tumor context. So now why suddenly ECM thing came into our interest area is because when we took the triple negative breast cancer cells and targeted UBR7, we observed that there are multiple processes that get deregulated, but one of the fundamental process that was found to be differentially altered is extracellular matrix organization. So this was kind of very interesting to us. And again, if we kind of do functional annotation to the pathways and groups, we find the TGF beta signaling cascade was definitely affected when you target UBR7. So now, what we observed is we did unbiased targeting and tried looking at the different epigenetic modifications. So it was a H2B lysine 120 ubiquitinating enzyme. However, in the TNBC context, in the endogenous factor being targeted, we did not see an appreciable change in the K120 ubiquitination. In contrast, we observed histone H3 lysine 27 trimethylation, which was globally affected when we targeted UBR7. And following this, when we went ahead and did chip sequencing analysis, again, you can see the 27Me3 levels were globally downregulated, affecting several genes and pathways which affect ECM. So this is shown for simplicity the TGF beta 1 gene, and you can see that when you do not have UBR7, the 27 trimethylation landscape is hugely downregulated, indicating that UBR7 is involved in actually regulating this ECM gene through some upstream pathway. So we performed IPMS study, and we took normal proliferating cells, MCF10A, and triple negative breast cancer cells, MDMB231, and we identified the interactome. And one of the particular complexes that hit our attention was this PRC2 complex, EZH2, EED2, and SUS12, while UBR7 did not interact with these protein complexes in the context of MCF10A. In the MDMB231 cells, we could actually get this entire complex protein isolated. So this was very interesting to us, and so this kind of get the model that we were thinking about is in the TGF beta gene promoter, UBR7 is definitely there, and it is instrumental in recruiting the PRC complex. So if you do not have UBR7 in the system, the ED2, SUS12, VISA-H2, all these subunits actually get dislodged. So we now hypothesize that UBR7 could be a formative complex component, which is very crucial for the PRC2 recruitment to chromatin. And you can see here that if you target UBR7, the EZH2 recruitment through chip assays was dramatically compromised. And so if you do not have UBR7 along with PRCs, TGF-beta activation happens, leading to activation of phosphosmad cascade, which turns on these ECM genes, collagen, fibronectin locks, and that is the signaling mechanism that we actually established. So now if you move from an adherent cell to a 3D model and you have UBR7 removed from the system, you can see fibronectin actually getting kind of hyperexpressed along with the LOX enzymes, which are the cross-linking enzymes. So these are the sphere formation assays, and you can see the total collagen content like that. We estimated fibronectin and other factors of the ECM. You can see there's an upregulation of these, the staining of these in the 3D context, showing the ECM is more robustly forming when you do not have UBR7 in the system. So from the tumor spheroids, we now estimated the LOX activity assays, and so you can see from the spheroidal cells as well as from the spheroid supernatant, you can see targeting UBR7 leads to an activation of the LOX activities quite robustly. And LOX, as I mentioned, is the enzyme which cross-links collagen and makes the ECM bundling more stronger and kind of more rigid. So now we went ahead to the mouse models and we targeted UBR7 and again complemented back with the wild type protein. And you can see the LOX activity as well as the total collagen content shows a differential expression when you don't have UBR7 in the system versus when you complement again wild type, it again reduces. So from the cells into the animal model, this actually works. And so these are the type 1 collagen stained tissues that you can see the similar way with the increase in the expression of the UBR7, of the knocking down of UBR7, the expression of this type 1 collagen fiber was distinctly increased in this context as you can see from the images. Then we went ahead to look into the patient samples and we did total collagen assay as well as LOX activity assays, and these are some of the kind of studies which clearly shows a kind of anti-correlation where UBR7 expression and LOX activity or total collagen content was inversely related. So these are the tumor microarray sections and showing some of these slides. You can see the kind of anti-correlation I'm talking about. Expression of UBR7 was much low, and then fibronectin and collagen was heightened in the tissues. And so there was a stage grade correlation. This was more prominent in the higher grades of tumor, that you can see this kind of anti-correlation functional. So finally, the context of matrix stiffening that we are talking about, we wanted to understand it in the cellular context, how we can score these assays. And one of the best ways to study the stiffness is doing atomic force microscopy. So here we cultured cells and then we looked at the native matrix formation, and the stiffness of these matrix was actually measured by AFM. So from the results you can see here, the stiffness was measured through DMT modulus, and you can see there is at least around 3 times the increase in the cellular matrix stiffness when you just do not have one factor UBR7 from these cells. So these questions could be all in vitro and might be all kind of some kind of data which we might not extrapolate in the cells, from the cells to the actual tumors. So shown here is a mouse tumors, and we again repeated our experiments where we did from the tumors the AFM studies, and you can see matrix stiffness is quite pronounced when you have this ECM overexpressed, and when you again co-overexpress ECM, the UBR7 along with these ECM proteins, you will see that there is a downregulation of this Young's modulus. So you can actually step it up, look into the animal model tumors, and you can actually measure the stiffness, which is beautiful, connecting it with the cell level data. Another small thing that I'll show here, but we are still working on this on much greater details. So we are looking at the cell-free DNA release from these matrices. So we know the stiffer the matrix, there's a release of the cfDNA is directly related to that. And so there are multiple mechanisms that are there in place, passive release models as well as active release models. We are looking into the active release models, which are extracellular vesicle mediated cfDNA release. These are some of the initial experiments, and we have done it in the mouse models also. And so what you see is control versus ECM overexpressed condition. CFDNA release is much higher, and again, you have co-overexpression, you can reduce it back. So this CFDNA release, people say, is some kind of intermediate step which decides the metastatic potential of the tumor as such. So to understand that, we looked at the invasiveness of the tumor. So these are tumor invasion assays, and you can see that LOX overexpression, the invasiveness increases. Again, co-overexpression with that, we see that there is a downregulation. So that we can connect the dots and say that there is a direct regulatory role of UBR7 in the context of matrix stiffening and downstream the invasive functions that you can see. So a part of the study that I have talked about is just published, and you can see here the TGF-beta is a master regulator, how it is co-regulated by UBR7 and PRC complex, and that kind of suppresses the activation of the ECM genes. In absence of UBR7, the PRC complex recruitment is not happening, leading to TGF-beta activation, and downstream there is activation of these ECM factors which are involved in directly leading to the alteration of the matrix stiffness of the cancer cells. So yeah, that's all. So these are my grad students, and some of them were involved in ECM projects, some of the metabolic regulation project, and these are my collaborators without whose help and continuous support, some of the experiments which are very challenging would have been difficult. And this is my project funding, and thank you for your attention.
Thank you. Questions?
Very nice talk, thank you. I just have a question about CMYND8, and like a few years back, it was shown by Michaela Divergilio's lab that it loads onto enhancers, and it has a role in transferring RNA polymerase from enhancers to promoters. So if you take it out, you get polymerases more loaded on enhancers and promoters. Do you see that in your studies in context of enhancers surrounding the genes that you are studying?
Perfect question. Like there's a super enhancer activation model that those group has shown, and since we have the chip sequencing data, we actually see in the kind of cancer context, particularly the core tumor context, there are lots and lots of non-coding RNAs and the enhancer mediated regulation which are coming up. So, but it's like a dots in the puzzle. We have to connect it and try to see how this entire thing fits into the magnanimity of the cancer. But yes, it's a very important happening event, and non-coding RNA regulation is in the forefront in this context.
Thank you.
Regarding the first part of your talk, I was curious if, because there is also metabolic changes, like the level of acetyl-CoA, I guess, are changing. So, and of course, this is very important for gene activation. So do you see crosstalk between the two, and how's it working?
That's a very wonderful question. So acetyl-CoA and the lactyl pools are kind of inversely correlated, and this guy is majorly involved in the control condition regulating acetyl-CoA, and in the kind of hypoxic condition regulating the lactyl-CoA levels. And that we hypothesize kind of has an important condition in the acetylation, lactylation of histones. We are trying to work more on that regards, and how this guy can switch the windows and promotes and that transfers the gene expression. But it is something to think about, that acetylation as well as lactylation of histones, both are transcription activation kind of context, but it's very context dependent event, early phase versus late phase. So it's all time dependent in kind of dynamics that is going on, something which we are very much interested in, and we are exploring that at the present moment.
Hi, nice talk. For the first part of your story, is it only specific for ER positive breast cancer cells?
Okay, so we have been looking at the different etiology of the disease, like TNBC. We have one set of study, and the one that metabolic regulation stuff that we are doing is in ER positive tumors, because of the fact that is more happening in the population, and the ER positive tumors and HIF1 alpha correlation is very nicely can be done. So the complex recruitment onto many genes and in cancer enhancer recruitment models that are there in place with us now. So that shows that in ER positive tumors, this is a very important event.
Is there any sequence specificity for the
Z-bindate? So see, it's a PBP containing transcription factor like PWWP, bromo, and PHD domain. So together it gets recruited to certain epigenetic sites, certain histone PTMs it prefers. But saying that there are certain DNA sequences inherent in the chromatin where it goes and binds. So it's not that everywhere the chromatin marks are there, it will go and bind. So there is a contribution of the DNA. That's why the enhancer activation model that we are proposing is there.
Thank you. Any other questions? If not, let's thank Chandrima for the nice talk.
Thank you.
Thank you. Thank you for the kind introduction. And first, I would like to thank Dimple, Ranjith, and Mithun for this wonderful opportunity. And thanks for putting together the workshop and the conference. So I have been learning a lot. Hope this will continue in the following years. Yeah, I'm learning. So it's good, both ways. So in this talk, I will try to explain some of the stuff that we are doing in the context of how chromatin structure helps in the cancer evolution. Okay. Just for the change, I put a different slide because everyone starts with the chromatin structure. So please wait for that. So we study cancer evolution. As we know, cancer is a genetic disease. It starts from the normal cell through the accumulation of some genetic alterations that provide the cells a growth advantage so that it can go and expand into more clones. Although we use tumor as a single entity, if you zoom into the tumor tissue, we see cells that are highly heterogeneous. Although all the cells are connected here because they share some same genetic material because it's all started from single founder clone. So you could see some similarity between them in terms of some genetic aspects. But as these cells proliferate more and more, they can accumulate. Is it not audible? Okay. Yeah, thanks. It's mutated. Now it's been fixed. So when the cells go on proliferating, they can accumulate new changes. And because of this, they might show difference as compared to the founder clones. And some of these changes might provide selective advantage for them to disseminate to the other cells, other tissues in the body, and then they could establish the secondary tumors, which often call it as metastasis. So although a lot has been known in terms of how cancer evolves, we still have a limited understanding of the source of these events. Because for all these events to take place, we need a continuous source of genetic and epigenetic alteration taking place. The genetic changes could be changes in the DNA, and the epigenetic changes could be modifications in the DNA or in the histones. And these changes could be accumulated because of the changes that is happening in the cancer cell with respect to the environment that it is facing, as we have seen in the previous talk. So our lab is broadly interested in understanding how these genetic and epigenetic changes are accumulating in the somatic cells, given that we know the genome is not just linear. So in the last few days, we have been learning that the chromatin is highly compacted and it's been assembled in multiple distinct structural units, starting from just a single-strand DNA, double-strand DNA, and that is being bound by some histones at the local level to form nucleosomes. And these nucleosomes assembles, and they can also mediate long-range chromatin interaction with the help of CTCF and cohesin that forms these kind of loops. More loops forms these tags, and then you can have a large-scale domains such as active and inactive domains. So the question we are asking is very simple. Given that this structure is highly complex, how does it let the DNA more accessible for genetic and epigenetic changes over a time? So how do we study this? Since we are a bioinformatician, we start with the data that is publicly available. So we use the data directly measured from the patient sample. Consortia such as TCGA, PCOG, and others have done a lot of extensive data generation from the patient tumors directly. So we know the somatic mutations from the patient tumors. We also have other set of data that has been generated from the same tumor. And from this, we are trying to look for patterns. The kind of pattern we look is, first is the mutational processes. Every DNA damage has some specific nucleotide context to be targeted. So by looking at the mutation frequency across the genome and where in the context it occurs, we have an idea of which mutations are more frequent and which trinucleotide or 5-nucleotide is more probable to mutate. So this gives an idea of what mutation processes are active in the tumor. And then out of the several mutations that we see in the genomes, which mutations are cancer driver? For this, we use a method called signals of positive selection, meaning if a gene is involved in the particular phenotype, we often tend to see that gene to be mutated across multiple individuals. So we are taking the information of the recurrence, meaning the same gene mutated in multiple individuals, as a proxy to say which ones have a likely driver events. And of these events, how many of these events are driving the tumor? And given that there is a lot of mutations happening in the somatic cells and also some driving potential, how does this tumor cell survive in the context of the immune environment? Because all this mutated protein can express some new antigens, and this could be targeted by the immune cells, but the cancer cells are smart, they find an effective way to escape from this. We are trying to study all these factors from this state, which is the advanced tumor in which most of the data has been generated. So I have this information. Can I use this to trace back in the context of cell of origin chromatin structure to ask where in the genome all these events are taking place? So today's talk, I will focus on a couple of events. One is the mutational process and cancer drivers, and I'll just give you how we can deduce these patterns in terms of chromatin structure. Starting with the mutational process, we have a lot of chromatin information from consortia such as ENCODE and Epigenome Roadmap, where they have profiled different epigenetic factors as well as the DNA accessibility in the normal tissues as well as the cancer cell line, so that we can have some broad idea of which of the chromatin regions are accessible in the normal tissues and also which transcription factors are binding. By taking that information and comparing to the mutation rate that we see in the tumor genome, what we found previously as part of my postdoc was that the mutation rate are highly variable across the genome. In particular, in the open chromatin regions where the DNA is bound by transcription factors and histones, we tend to see more mutations accumulating at these protein-bound DNA sites, especially in melanoma and lung cancer, because both of them had a high exposure to the environmental factors such as UV light or tobacco smoking. Both of them creates a large distortions in the DNA helix, and because of this, we need DNA repair such as nucleotide excision repair to fix those damages. So what we hypothesized was that perhaps the presence of the DNA bind protein could hinder the accessibility of this repair to those damaged sites, and because of this hindrance and in the next round of replications, some of these damages could turn to a mutation, especially in the cancer cells. This could happen more rapidly because of the compromised DNA repair. As a result, you would see more mutation at these sites because of the low repair at these sites. Okay, so this is fine in terms of melanoma and lung cancer, but at the same time, we have observed something else where this particular protein-bound sites like CTCF and cohesin, which are at the chromatin loop anchor points, they seem to be mutated across multiple tumor types. Not only in melanoma and lung, it's been seen in other gastrointestinal tract cancers, colorectal, gastric, and esophageal. However, the molecular mechanism underlying this is not clear. It's not simply because of the reduced nucleotide excision repair. So this is the first problem we started working on. While going through the literatures, what people alluded is that the higher mutation rate at these sites could be explained by challenging DNA replication, because when both the proteins are present, that could create some torsional stress and so on. And they often found that we see more mutation at this site, especially when the samples have high genomic instability. Taking this information and then some preliminary computation analysis, Fasila, the first graduate student in my lab, come up with an hypothesis that perhaps CTCF and cohesin binding to the DNA during the replication phase could create a torsional stress, And because of this stress, these sites may undergo break. And if those breaks are not prepared or fixed well, this could be the source of mutation. So this is the hypothesis that we wanted to test. Of course, it needs a lot of data generation as well. Thankfully, we are close to Dimple, both in terms of physical lab as well as in terms of the project wise. She helped a lot in setting up this experiment to test this. So first thing we have done is we want to see whether CTCF and cohesin are bound to the chromatin during the replication phase. So all the data set that are available in ENCODE and roadmap are done during the asynchronous condition. And with that, we don't know how many of them are actually bound as a part of the replication phase. So for that, we have taken the HeLa cell line, which was available, and we synchronized them to the S phase, and then we have performed the chip-seq of CTCF and RAD21. RAD21 is the part of the cohesin complex. And by doing the chip, and we could see that there are many genomic locations where both CTCF and cohesin are bound together across the genome. We have around 34,000, of which 26,000 had a strong motive for the CTCF. So we'll be using this for the rest of our analysis. And first thing we asked that, are these sites specific to the S phase, or it's been shared with the interface as well, which is the transcriptionally active state. And what we found was that almost 80% of them are overlapping to the transcriptionally active sites or active state. Not only with the HeLa, it's also overlapping with many of the asynchronized population from multiple cell line, suggesting that many of these CTCF cohesin binding site that we find are persistent across multiple cell types. Okay, if these two proteins are bound to some extent, does it affect the replication fork progression? So that would be an obvious next question we can ask. If you want to do those kind of analysis to check if the protein is stopping the replication, people often do a replication dynamics assay or the fork progression assay. But in this case, we want to study how this fork is being stalled at a particular focus. So for that, replication dynamics assay is a bit difficult. So we focused on some of the protein that could be used as a proxy to measure the replication stress or the replication fork stalling. Some of the proteins involved in this one are STN1 and MR11. For example, this is a roadblock for the replication progression. If there is a roadblock, these proteins could come and help to rescue this region to avoid any fork collapse. This RAD51 decorates the exposed region, and then the STN and MR11 prepare these regions for fork reversal, and then with the help of homologous recombination, these regions would undergo proper replication. Okay, so we have been tapping onto this protein to see if there is a fork stalling happening at the CTCF cohesin. So we have done the chip-seq for the MR11 first at the mid-S phase, and we have plotted the intensity of that chip-seq signal at the center of the CTCF cohesin binding site, like 20,000 odd CTCF that we have found from the mid-S chip-seq. And what we are seeing here is that there is a strong signal at the middle of the CTCF cohesin binding site, or so-called CBS. And this signal intensity is higher as compared to the immediate flanking region, suggesting that it's happening more often at the closer to the protein bound sites. In order to check if this signal intensity is very specific to the CBS, we have taken some control sites which are CTCF unbound. We have just the motif, but there is no chip signal for the CTCF. And we also selected some random region, but in those cases, we don't see as strong signal as we can see in the CTCF and cohesin binding site. Similar signal has been seen for the STN1, which works together with the MR11, so we can see a replicate of that. And we also have seen the signals for the break as well at these sites, which is often measured using the gamma H2X, which decorates the nucleosomes that are nearby to indicate that there is a DNA double-strand break happen at those sites. And then, of course, if the strand is break, some of the exposed sites needs to be protected. So we have the RAD51, and those signals are also enriched at the CTCF cohesin binding sites. Collectively, this tells us that the CTCF cohesin binding sites are undergoing a lot of DNA breaks. Okay, if the breaks are not prepared or fixed well, this might lead to more genomic instability, right? We can test this by taking cell lines and knock down this STN1 or MR11 and then see what kind of genomic instability it could accumulate. An alternate to that is we can tap onto some of the genomic sequencing data from the patient samples which have deletion in one of these genes and then ask how the mutation rates are accumulating at those sites. So we have used the pan-cancer analysis of whole genome, which I was also part of previously. So from this, we could segregate the samples that have both MR11 and STN1 wild type, and then MR11 or STN1 deletion or both deletion. And then we looked at the mutation rate at CTCF and cohesin. So on the left is the mutation rate plotted at the CTCF and cohesin corresponding to the x-axis 0, and then you can see how does the rate compare to the immediate flanking regions. So in the wild type, we still see some level of mutations at this site. However, when the MR11 and STN1 is deleted, we could see a stark increase at the mutation rate at the middle of the CT7 cohesin binding site, suggesting that if these genes are compromised, these sites will never get fixed and tend to accumulate more and more damages. And this increase was not seen in case of controlled or CTCF alone or RAT21 alone sites, suggesting that CTCF and cohesin is an important factor in increasing the mutation rate at this site. This is the case for esophageal. We have seen the similar trend with other tumor types as well, suggesting that this is a common mechanism seen in multiple cell types. Okay, in summary, we know that CTCF and cohesin binding are involved in multiple function in terms of the chromatin looping and gene regulation. If some of those sites are bound still during the replication, there might be a roadblock, and there could be torsional stress happening because when you are unwinding the DNA and then you have a stopper there, all the stress will accumulate at particular site. And usually this stress will be reduced by the TOP2B or other topoisomerase, and then it can go for a release of the stress, and then the homologous recombination repair would come and fix those sites. But unfortunately, in the cancer cells which have a lack of this STN1 or MR11, these sites are not fixed, and this become a source for more mismatch introduced at these sites. And because of the immediate rebinding of the CTCF to these sites never been fixed by the mismatch repair and become the source of mutations later on. Okay, it's a two-part thing. The CTCF and cohesins are essential for the DNA break. And if the CTCF bound back to a mismatched region, this never get repaired and it become a source for the next round of mutations. Okay, so we have more details in this preprint if you're interested. And I'm going to the second part where I'll talk about how these mutations could affect the gene regulation. So in the first part, we understood what are the processes that could cause the DNA damage at these sites or the mutation at these sites. And now can we ask how does that affect the gene regulations? So that is an ongoing work that we are taking to study how the mutations at the promoters or in the CTCF cohesin sites that are in the intergenic region could affect the gene regulation by having a direct cis effect or a long range effect. And we also study the effect on certain transcription factors like P53 and MYC, which often gets mutated. How does that affect the chromatin landscape and the gene regulations and so on? For today's talk, I will focus on the cis effect, and that to a particular form of the DNA damage, which is introduced by a foreign particle, which is the virus. So in some tumor types like virus, it's often associated with the infection called papillomavirus. Papillomavirus is the DNA virus, and it infects the epithelial cells of the cervix. And most of the time, our body's immune cells will have a capacity to recognize the foreign entity and clear off. And this is especially boosted if you are given vaccination for the HPV. Okay, so the main part of the HPV vaccination is to boost our immune cells so that whenever it encounters this particle, it gets cleared off. But in unfortunate situation, if any of this viral particle enter the basal layer of the epithelial strata, which is often the source for many of the cells that we see at the top, the virus will take advantage of these same cells and then they replicate their own copy as well. And since this is the DNA virus, it also has some genes that the virus uses. Two of them are E6 and E7, which are known to perturb the host cellular pathway as well, which includes P53 and RB. And because of that, the cells would have a growth advantage and it became more progressive at this phase. In many cases, even this condition is also be regressed because of body's immune cells. But in unfortunate situation, some of these cells might transform to be invasive. So during that invasive transformation, what happens is that this episomal circular DNA can also get integrated to the host DNA as well. And the way it happens is that if there is a break in this genome as well as in the host genome, because of some shared microhomology between the virus as well as the host gene, the microhomology mediated end repair can fuse this extra chromosomal DNA to the host DNA. Okay? And this integration can happen random wherever you see some shared microhomology between the two genomes. So what we want to understand is that how much this integration is favoring the cancer progression. This is important for two reasons. One, we often see the integration in the advanced stage tumor, and two, it's always been associated with the clinical response as well. So if you segregate the samples which have just episomal form as well as the integrated form, the chances of them to develop recurrence is much more if the patient has the integration in the tumor as compared to the episomal form. And higher the integrations, there is a higher chance for the recurrence and the overall survival. So it's important to understand where this integration happens and what is their function. So previously, as I said, a lot is known about from the genes that are expressed from the viral DNA, but not known is about the impact of the integration itself. Because we have been studying about the chromatin structure, how much it is important for the gene regulation, and now you can think of an integration that comes from somewhere, which has the genome of size 7 kb or 8 kb. So this integration often is a size of 7 to 8 kb. If this DNA gets integrated to the host DNA, how much is affecting the host chromatin structure is what we are studying in general. Okay? This does not just come as an integration. We often see the integrated have some regulatory region, and we know what is the strength of the viral promoter when we are designing the plasmids, right? So let's say if this integration comes with the promoter, how much that promoter is going or enhancer is going to affect the function of the nearby genes where the integration takes place. So this is what we want to study. And for that, we have taken the integration information from the patient samples, from the whole genomes, as well as the hybrid captures. And then we try to compare the integrations in the context of the chromatin information from the closest cell line possible, because we don't have the chromatin information from the patient tumors, the matched patient tumors. So in the interest of time, I'll just focus on what are the main findings from this analysis. The first is we asked if I have an integration in a particular TAD, how does the expression of the nearby genes looks like? And how does the expression of the genes that are in the 5' and 3' TADs as well? So at the middle is the TADs that have an integration, and in the y-axis we see the average gene expressions that are present in the same TAD. And what you are seeing is the samples that have an integration have a slightly higher expression as compared to the samples that don't have integration, but the gene expressions are coming from the same TAD from other samples which don't have integrations. And you can see the difference. There is an increase in the expression when the integration happens in the TAD. So what happens to the neighboring TAD? And interestingly, we find that the neighboring TADs are mostly unaffected, meaning the insulation that we have been seeing so far is acting here as well, that prevents the activity of the viral integration to the adjacent TAD and confines all their activity to the TAD that have the integrations. Okay? So now we zoomed into these TADs that have integration and asked which genes are in those TAD and how many individuals have integration in the TAD. So what you are seeing is a heat map. The x-axis is the samples, and here on the y-axis is the different TADs. We just numbered to differentiate between them. And here the darker blue indicates there is an integration from that sample in that particular TAD. And these are all the TAD that have most recurrent integrations, and you can see very nicely that there are many individuals that have integrations in this TAD, followed by this TAD, and so on. And also you can see nicely there is a mutual exclusivity, meaning that not many samples have integration in more than one of these TAD, suggesting that perhaps a single or a multiple or a couple of integrations is sufficient to drive the tumorigenesis process. We looked at what genes are enriched in this. Of course, the TAD that have a high number of integration has the MYC and PBT1, which is well known, as well as TP63, RAD51, and so on. So then we asked how does the integration affects the expression of this gene within those TAD. So for that, we have segregated the samples that have integration and don't have integrations, and then looked at the expression of the MYC, PBT1 that are present in the same TAD. And as we have previously seen, there is a higher expression of these genes if they are integration happening in the same TAD. Then we looked at one particular gene, MYC, which is most recurrently mutated, and asked where the integrations are happening in the context of the gene position. In the x-axis, the 0 corresponds to the start of the MYC gene, and each row is a sample. And what you are saying is how the integration are spread with respect to the promoter region. We could see in many samples the integrations are closer to the MYC gene. And you see many dots because the integration is not always precise one integration. It's always integration followed by multiple copies of the nearby integrations. So we often find multiple integrations nearby. And there are samples that have integrations that are really far away, but within the same TAD. So then we try to look how these integrations could affect the MYC expression. If it is nearby integrations, the overexpression of the MYC could be explained by the run-through of the higher activity of E6 and E7. If I have a strong expression of these viral genes, some of these polymerase could run through of the downstream genes so that it can increase the expression. If it is the long term, so the long range, we thought that perhaps there is a looping involved by connecting this HPV integration to the MYC that is downstream of this. To test if this is possible, we took the HeLa cell line, which also have a HPV integration in chromosome 8, and that has already been shown to have some kind of chromatin interaction through the chia PET. But to check if this is happening at the lower resolution, we have done 4C with the help of Dimple lab, where we have designed primers on the HPV integrations or the HPV genome and asked what other regions within the TAD are interacting with that. And we could see much of the interactions are happening between the HPV integrated region as well as the MYC and the promoter region. Okay. If there is an interaction happening, it needs at least some of these components like CTCF and cohesin. What happens if you knock down that? So we have used the data from this paper where they have done an AID-mediated knockdown of CTCF and cohesin separately, and they have performed promoter high C. So when we took the data from this analysis, we found there is a strong reduction in the chromatin interaction when cohesin is knocked down as compared to the CTCF. And in relation to that, we also see a decrease in the MYC expression as well, suggesting that the chromatin looping is mediated by cohesin between the integrated site as well as the MYC promoter. So this is the model that we proposed. In the initial stage, it exists as an episome, but over a time, it gets integrated and then drives the expression of the oncogenes as well. And that's why we often see the recurrent integrations closer to the oncogenes in the patient samples. Okay, this is something to understand. Is it useful in terms of the translation? We are now taking this information at the level of the screening. For example, if somebody goes for a pap smear and been observed with the abnormal changes in the cell, we suggest them for, or we recommend them for HPV testing and the integration analysis. And the way we do that is we do a low-pass whole genome sequencing or nanopore-based sequencing to find where the integrations has happened and whether that integration is seen in one of the TADs that we have seen with recurrent integrations in the advanced stage tumor. By combining these two information, can we tell upfront how likely an individual will develop or have a higher risk to develop into an invasive tumor? So this is work in progress with people from my lab as well as some collaborator from the KMC Medical Hospital. Just skip the summary slides, but just to mention that although the genetic alterations are the founders of the tumor evolution, we think that the progression, metastasis, and resistance could be driven by the plasticity of the tumor cells rendered by the chromatin structure. Any changes or the flexibility for the cells to happen, it should happen at the chromatin level, and we are trying to study to what extent the chromatin structure is feasible for that. As a part of this effort, we are also trying to do a single cell multi-omics to understand how the chromatin structure and regulations are changing at the single cell level. And this is carried out by Dr. Fasan, who is here in the audience. We also have a poster explaining some of our initial steps in doing that in the head and neck tumors, which is more prevalent in the Indian context. With this, I would like to thank Anurag and Fasila, whose work I have presented, and rest of the lab members for their support, collaborators, NCBS and India Alliance for funding, all the consortia who has made the data publicly available with which we can develop all these things. I'm sorry for the extra minute, and I'm happy to take questions. Yeah.
Very nice talk. This integration is random. So is there a high chance that it will be near an oncogene, or is it be low probability?
Yeah. We think in the initial phase it could be random, but if the integration is closer to any of the oncogene, that might have provided selective growth advantage. And since we are looking at only the advanced stage tumor, we often tend to see the integration closer to the oncogene because those are kind of selected or the fixated one. But the initial stage, it could have been random.
Okay, Shabari, very nice concept. My question related to your first part. So you mentioned the topological context. Instead of that, what happens where there is a loop, the top two always sits at the loop, right? So whatever you're reading as a damage, maybe it's caused by the top two itself to release that, right?
Yeah.
So do you still see this damage if you inactivate or reduce the function of the top two or inactivate the top two?
We tried, but the problem for us is to connect to the mutations at the later stage. But if you reduce the top two, or first we tried the chipsake of the top two, although it didn't work very well, but we could see there is an enrichment of the top two at these sites. But that's something that we could discuss and follow up.
Okay, thanks.
Have you specifically seen if there is these CTCF-driven mutations is there any bias for these promoters which have CTCF binding sites in the vicinity?
Yeah, so we are working on that, but we see mutation mostly in the sites where there is a plenty of CTCF clusters. So even if one is mutated, perhaps the other sites are behaving as a buffer to still continue the function. But there could be one or two CTCF definitely driving the cancer progression, but we have to find out those.
Is there any bias in where it is happening?
Yes. So it is at the 9th position where we have a big A. So somehow mutating that is not affecting the binding strength so much.
Yeah. I didn't quite understand the model with MYC and HPV insertion. Can you go back to that slide? I just had a question about it. So when you, yeah, right there. Well, go. No, the one where you show the integration sites with respect to where there. So
this one?
Well, just without the cartoon, though.
Okay.
So when you have, say, insertion of HPV at that distal enhancer, say -400, -500, so your model is that it's activating that enhancer or inactivating it?
Activating that.
And it's activating it just by getting transcription through that?
Yeah, we think that HPV itself has some regulatory regions, and this could recruit some transcription factors. And because of the looping between that and the promoter region, there is more transcriptional activity happening at the MYC.
But the model is activation of that enhancer
it's an activation.
by insertion. Okay,
yeah.
Thanks.
Most of the time it's an activation because it comes with the additional regulatory region. Okay. Yeah. And people have shown in other tumor types there is a lot of rearrangement happening upstream of the MYC, and that is influencing the expression of the MYC. In the cervical cancer, we don't see so much breakpoints there, but rather these are all HPV-mediated enrichments there.
So, a very interesting talk. So I have a question about the breaks that you are seeing which are coercin CTCF dependent. So what I understand from the previous question is, Andre Nussensweig and others had shown that 2-isomerase 1 and 2 cleaves it and resolves it. And normally what you are indicating here is a homologous recombination-based mechanism, right? So in that case, don't you expect double-strand breaks occurring there in an insertion of structural variants rather than somatic mutations that you are putting in? Normally, if you see somatic mutations, you wouldn't expect gamma H2X foci because it's not a break, it's just mutations. But if it's a break and you get increased structural variants, you will get a gamma H2X. So have we looked for variants like insertions or inversions or amplifications there?
Fair question. We have gone through that paper. What they have showing is during the transcription, there could be stress happening and that lead to a break. We think that some of the break, if they are not fixed, they would still stay during the replication phase as well. That could be the source for the double-strand break. In addition, because of the replication also, that's what we are trying to add, that in addition to the transcription, replication also creates the stress, and there is a break happening. The break is often either single-strand or the double-strand break, and that's what we have capturing with the gamma H2X. If I look at the distribution of the structural variant from the patient sample, they are widespread because there is no specific sites where the break point happens. So if I see there is a high enrichment in the nearby regions, but there is no hotspot. Whereas with the point mutation, we see a hotspot there because the mutations are context specific. And that's why we are picking up this signal. But we are seeing the structural variation as well near that. And also another paper that connected that if I see more structural variants around that region, that is strongly associated to the hotspot mutation that we are seeing. So it's just if we show one, we can predict what the other things could have been happening.
So I'm curious about this torsional stress part that you talked about. So are there any measurements for that? Like can people actually measure these kind of, what is the magnitude of this torsional stress or something like actual biophysical characterization? Has it been done at all?
I think it's possible, but I'm not the right person. Maybe Maipal or somebody else could take that. Or in the interest of time.
One question, that's...
Okay.
Sir, I have some basic question. So in case of other viral integrations, for example, HIV, we're not seeing any kind of cancer progression. So is there any specific reason that HPV is having the cancer progression? Maybe the site specificity or something?
Yeah. Again, to my knowledge, it could be possibility of both. This HPV has this E6 and E7 that is expressed from that could affect the P53 and RB pathway. And that gives a stage for the cells to transform. And then the integration itself is also regulating some of the genes. So collectively, it provides a growth advantage. That's why we might be seeing this cancer association. And there are other viruses that are associated with cancer too, like HCV and others, EBV as well. But I don't know much about the HIV related things. Cool. Thank you.
Thank you, sir. So then,
am I audible at the back? Yeah. Okay, so hello everyone. Thanks Leelavati for the introduction and organizers for having me here. So I'll be talking about a histone modification and its role in interphase chromatin memory when the cell is dividing. So I'll not really spend much time on this because Sabri already introduced. But what I'd like to emphasize is that there is this hierarchical organization which is only true for the cells which are in interphase. And that is the period where cell spends most of its time because only like 5–10% of cells are there which are in mitosis. But interestingly, when mitosis is happening, the linear architecture is lost because you see the chromatin condensing, the hierarchical architecture is lost. What you see is that the chromatin is now linearly condensing such that the rod-shaped structures are formed, and now you don't see any chromosome territories, domains, or tags. So the question is, when this is happening, the transcription as well as the structure is lost during mitosis. How do the cell retain that? Because it's very important that in multicellular organism, the cell type specificity is maintained. So what happens is, if you consider a cell as a book and there are different pages which are genes, and you want to remember which gene has to express in that cell, what you would do is you would bookmark this. So this is called mitotic bookmarking, where you keep the transcription factor bound or certain histone modifications while most of them are being removed to maintain fast transcriptional activation in the next interface. And this field is still growing, but we do have certain idea about how enhancers and promoters are retained in this context. The problem comes when you want to retain the structure. So if you look at this heat map from Decker's lab, so they did this Hi-C in cells which were dividing. These are HeLa cells from, if you look at from early G1 to M phase. And then in a non-synchronous population, what you see here in these two plots is, you see beautiful AB domain alterations, which is lost in mitosis. You see again TADs, which are lost in mitosis. So the structure is not really captured with the Hi-C that we are seeing here. So now we ask, is there a way to retain the chromatin structure as well as function? And we hypothesize that this can happen with the histone modification because they are quite abundant. So I'll now acetylation and methylation of histones is being spoken about, where the retaining of these two. So acetylation to some extent, although it decreases, but methylation, which is consistent, people have spoken about how retaining those at enhancers and promoters can maintain some type of cell specificity. What is not really known is what happens in the context of histone phosphorylation. So we picked this modification H3S10P, and why we choose this is because it's quite abundant during the mitosis. As you see, it's the most conserved among the different phosphorylations. You see tetrahymena having H3S10P in the nuclei, the roundworms, Drosophila cells, as well as mammalian cells. So all the cells that are dividing have in mitosis H3S10P being accumulating on the chromosomes. So what we ask is, if the increase in H3S10 phosphorylation that you're seeing during mitosis, it can help in maintaining the structural as well as the functional chromatin memory in all dividing cells. So for this we choose HeLa cells because there's a lot of data set available that we kept mining for this study. So HeLa cells, if you see here, these are the cells which are mostly in interphase. Only few cells are in mitosis. To enrich the mitotic cells, we treat them with nocodazole drug. And then we do a chip sequencing because we want to look at the enrichment of H3S10P on chromatin. So what you're seeing here is from asynchronous to mitotic condition, there is this fourfold enrichment in H3S10P on the chromatin. And the obvious question is, where is it present? So it turns out that it's mostly around TSS and at the promoters. If you compare from asynchronous to mitotic, you see increase in the promoter regions. And the same can be appreciated from this snapshot of browser, where you are seeing acetylation mark, which is H3K27 acetylation. Now this is an enhancer mark as well as a promoter mark. And if you compare these two peaks, this is around MYC gene, again a highly transcribing gene. So you see H3S10P increasing here, but not really any enrichment here. So, okay, in bulk we see more at promoters, but not in enhancers. But this was fine. What caught our attention was that once you start zooming out of the on the genome, you start noticing these domains of H3S10P. So H3S10P is forming these large size domains, and few are shown here by this arrow. So this is chromosome 11 from start to end, and you start seeing these domains. So next we asked, okay, do they overlap with any of the interface structure? So interestingly, if you take high-C data from interface cells in HeLa, A and B domain, what you're seeing here is that the H3S10P domains are overlapping with the active domain. Now remember, these domains in mitosis are completely removed, as I just mentioned in the introduction. So you are not seeing any A domain or tag in mitosis, but still you are able to have H3S10P on those domains. This is quantified here. So B domains don't really enrich much of H3S10P, but A domains do enrich. So what we are saying here is that in dividing cells, here it's HeLa, but we have checked it for another like different cell lines. So that in mitosis, the structure is not there, but the phosphorylation is increasing on the domains. But this was about the physical enrichment. What about the activity? So it turns out that if you divide these domains, now these domains are again from interface, and we are dividing all the active domains based on H3S10P enrichment. So you are seeing high H3S10P, medium and low. So there's a grade of H3S10P in these domains. And if you go back and ask from the asynchronous population, so which means in interface, if you take the H3K27 acetylation mark, which is again an active mark, you see the same grade of active mark deposition on these domains. So what it turns out is that H3S10P is not only physically marking these domains, but it is captivating the function as well. So along those lines, we also check the tags which are present in these domains, and it turns out if there's more activation, you will have stronger tags, stronger interactions, which is captured with this plot. So the higher the H3S10P, more the interactions and activation. Okay, this was good, but what is exactly H3S10P doing there? So there's literature that suggests that H3S10P might be involved in chromosome condensation or local compaction, depending on the context. So what we asked is, again, we took the Hi-C data from Decker's lab, who have done Hi-C in mitosis. And what we are doing here is, I would like you to just focus here. So we are taking all the TADs which have highest NP, lowest NP, or which is a random category, and we pile them up and then check the enrichment, intra-TAD enrichment. So you can see here, the blue color indicates more intra-TAD interactions and the red is the opposite. And the same is captured in this plot, which is distal to local interaction, again from the Hi-C data, where what we are seeing is that locally the TADs which have higher H3SNP are more compacted, and this is the data from mitosis. So okay, I'll just summarize this here. So in interphase, there is a structure, there is inactive domain, weak domain, and active domain. In mitosis, that structure is absent physically. H3SNP is deposited in a way that it recapitulates the activity of these domains when it's increasing in mitosis. And not just that, it is compacting the domains where it is more. So you are compacting the most active domain the most. Okay, but then the question was, what exactly is H3SNP doing? So if we deprive the cell of H3SN phosphorylation in mitosis, what will happen? So what we do is, we make stable cell lines in HeLa with this construct. So we have HA tagged with HA tagged with H3 serine 10, and the serine 10 mutated to alanine or the aspartate. Now both of these mutants cannot get phosphorylated, but one is it doesn't carry any charge, and the other one has the charge. Okay, so the system is such that it's inducible. Only when we give doxycycline, the mutants or the variants are going to express. So what you're seeing here is, once I give doxycycline, the cells have the HAH3, otherwise they won't have. And the beauty of this system is, once you have a set timeline of arrest and release, because we want to study that in the mitosis or the next interphase, how the absence of H3SNP will have any effect. So what we are doing is, only just before the mitosis that we want to capture the data, we are inducing these mutants. So each cell line will express these different mutants, and then we ask what is happening to the compaction in mitosis, and in the next interphase, how the function or the structure might be changing. So for this, the mutants, what they show is, if you compare the wild type with the A and D mutants, this is metaphase spread. So you're seeing the chromosomes are more elongated, so probably the compaction is now less, and because of which you see that there is a compaction defect. And it turns out this compaction defect is nucleosomal. And why I say that is, if we did H3 chip sequencing, which is standard in the field, so when you want to look at the mitotic compaction, you would do H3 chip seq, and you can appreciate here that although the differences are subtle, but you see that there is decrease in H3 incorporation in the domains that have high H3SNP. We check this for promoter as well, so which means that locally at the promoters as well as on a large scale at domains, even on the chromosomes, you are seeing a compaction defect in mitosis again. Now the question was, in the next interphase, what's happening? So for this we did ATAC-seq, where we are looking at the chromatin accessibility. Now the data that I haven't put here is, when you divide the genes based on their expression, you see H3SNP being highest on the highly active gene. And so we divided the genes based on their expression from low to high expression. And we are seeing that, again, this is subtle, but you see that there is increase in the accessibility in the next interphase. And this is the plot for the same. Interestingly, this is captured in the RNA-seq as well, because you are seeing that the genes which are highly expressing over the wild type, they are gaining some quotient of transcription. So what we are trying to say here is, H3SNP is important for the compaction of the domain locally as well as for the large scale A domains. Once it's absent, meaning the mutants of histones get incorporated, the H3 occupancy is probably, the H3 is unstable, because of which you see that the compaction does not happen anymore, which leads to, probably, it leads to more of the transcription machinery being retained there. As a result, there is an additional quotient of transcriptional activity, or maybe the factors or the modifications, which result in this hyperactivation of the already high transcribing genes. So with that, I would like to thank Sachin, who helped me with the experiment, the analysis and the conceptualization of the story are Arif, Rajat and Anurag who are here. They helped me with few experiments and all the Dimple lab members for their help. Special thanks to SIF and NGS at NCBS who make these experiments possible. Shrimonta and Avinchal from ISC who helped us with the MESC culture. Funding sources, and if you would like to read more, this is the QR code. I would be happy to take questions now.
So, okay, so thank you very much for your interesting talk. Okay. So I'm wondering how much mutant, H3 mutant were expressed by induction? If you compare to the endogenous H3.
Right. So we have those plots, maybe I can show it later, but it is, let's say 1/5 or 1/6 of the endogenous one. So it's very less, but still we are seeing these defects, yeah.
Thank you.
Great stuff. I was curious why you don't see this phosphorylation on enhancers. If it is tracking the active chromatin, do you have any ideas on that?
Okay, I have like answers in two ways, not the exact answer. So this modification H3-SNP, it gets deposited by mediation of HDAC recruitment on the chromatin. So it turns out more the acetylation, more the deacetylation with respect to mitosis, which will lead to more H3-SNP deposition. So if you compare enhancers and promoters have it more. That's one guess that it might be more here. Other thing is there are few papers which have suggested that cells bookmark the promoters more than the enhancers. So maybe it's just adding to that part.
It's a nice talk. So is there an RNA component in this? Because people have previously seen that the RNA hybrids or R loops also accumulated a lot of H3-S10P phosphorylation, and it was related with condensation of chromatin at the pericentromeric region. So my question is that if you treat your cells with RNase H or overexpress RNase H, will it overcome the problems you face by putting in the phosphorylation dead mutant? It's a complicated experiment, but just I'm asking if there's RNA component in your biology.
So, yes, the R loop paper is there. We haven't really gone into that direction, but what we had tried is, if we were thinking more than any other thing, transcription, H3-SNP deposition is more correlated to transcription. So we had tried these experiments where we will treat the cells with DRB, which will stop the transcription, and you start seeing less of H3-SNP deposition. So there is an RNA angle, but exactly with respect to the condensation and in the mutants, if we can rescue, that would be a good experiment to be done.
You are focusing on the active parts, but serine 10 phosphorylation is also Q1 binding.
Right.
Does it work? Yeah, so I was curious, so when you have your different mutants, what happens to the heterochromatin, and is it somehow also related to what's happened to the active part, or what's the crosstalk?
I was actually expecting this question because there's this phospho-methyl switch that people talk about. We surprisingly don't get much of H3-SNP on B domains. There are some B domains which I see in my data that will have H3-SNP, and they again are very strong. So maybe there is some association, but in the mutants or in the wild type, we don't really see heterochromatin changing or heterochromatin being marked by on a large scale. So maybe the time scale that we are picking, so we are picking the pro-metaphase cells. I think there is like maybe a dynamic factor to it, but yeah, that's there.
Okay, so
thank you so much.
Let's thank her again. Our next speaker is Shrita Chakraborty. She is at NIH.
Okay, so I'll begin by thanking the organizers for giving me this opportunity to present my work here. I guess I can continue speaking. So today I'll be talking about how CTCF loops restrict the range of enhancer action and how it impacts gene regulation and cell-fate specification. So we all by now know that genes are regulated by enhancers, and these enhancers can be located up to megabases away. And it is equally important to ensure that the enhancers do not contact inappropriate genes, and for this, insulators or boundaries play a very important role. And of course, it's not as simple as you see in the linear model, because thousands of genes in a 2-meter long genomic DNA is packaged into a nucleus of a diameter of just few micrometers. So DNA is packaged into these chromosomes that are partitioned into this megabase size domains with a high intra-domain interaction frequency and insulation in the neighborhood. Now, these domains are formed by the combined action of CTCF and cohesin, and by DNA loop extrusion, enhancers can come closer and physically contact the genes that actually promotes active transcription, or the CTCFs can act as insulators and they can repress gene expression. However, a few in vivo studies, including ours, have reported that some enhancers that regulate SOX2, for example, can bypass CTCF-mediated insulation, and loss of CTCF binding basically has no effect on SOX2 transcription. So what we asked is that what then defines the ability of CTCF loops to promote or block distal enhancers from interacting with the target genes? And for this, we focused on this high interacting domain, which comprises of the 3 FGF genes that you can see are demarcated by the CTCF clusters. Now, FGF3 and 4 have mutually exclusive expression pattern in the blastocyst, and FGF4 is critical in regulating the cell fate of the epiblast and the primitive endoderm cells. And later in development, all these 3 neighboring genes have very distinct tissue-specific expression pattern and also very distinct roles in development. So what we specifically asked is CTCF required to insulate the FGF genes, and can loss of the boundaries or CTCF cluster lead to ectopic FGF expression, at least in some cell types? So we started targeting this cluster. So from left to right, we numbered them as the CTCF motifs as C1 to C10. And when we deleted this 6 kb cluster within FGF3 and 4, we found that mouse development was not affected, despite the importance of FGF4 in regulating implantation. Loss of this motif, this cluster here, actually leads to perinatal lethality in homozygosity, and the pups die because of brain and heart defects. But most strikingly, loss of this C1, C4 boundary led to 100% perinatal lethality in a heterozygous background. And these pups develop encephalocele, where noodle tissue grows outside the skull. And this is the first report that we are aware of where we see such a severe phenotype upon loss of a boundary, even in a heterozygous background. So because this 25 kb deletion could potentially delete regulatory elements, and to make sure that the phenotypes that we see comes from the deletion of CTCF motifs, we actually made a genetic rescue line where we use the same guide RNAs that targeted this C1, C4, and now we used a repair template which basically replaces this 25 kb region with 700 base pairs of CTCF motifs, which are now surrounded by LOXP sites, which means that this line still lacks 24 kb of the region. And we found that homozygous mice was viable and fertile. And when we bred these mice with E2A3 lines, we were able to excise out the CTCF motifs, and we got embryos which recapitulated our phenotype, which clearly proved that the phenotypes that we are getting is because of loss of CTCF motifs. So now we can conclude that some CTCF loops, disruption of some CTCF loops can actually lead to extreme phenotypes. However, this is not true in all contexts. And so we asked what confers phenotypic robustness to perturbation of CTCF boundaries in some developmental contexts. And one good example is its enormous robustness during implantation. So what you see here is FGF4 is active in epiblast, and these are ES cells representing the epiblast. So you have this ES-specific enhancers, and you can see that in ES cells, FGF3 is repressed by deposition of K27Me3. So what we hypothesized is that loss of this insulator, we expected that loss of this insulator would be able to expose these ES-specific enhancers to FGF3 and lead to its ectopic expression. So the first thing we did was we did a region capture micro-C by loss of this boundary cluster between FGF3 and 4. And at a glance, you don't really see any change. But then if you look at the individual pixels, you can see increase in CTCF-mediated contacts across the cluster that comes from the two surrounding CTCF clusters. However, the FGF4 enhancer-driven contacts typically remain unchanged. And most importantly, if you look at the FGF4 enhancers, the profile looks very similar to the wild type, and this cannot spread out in the absence of the boundary. And in line with this, we saw that FGF3/4 transcription was not affected in early embryos, and there was no defect in implantation. So what we summarize from here is that boundary deletion between FGF3 and 4 can lead to loss of CTCF insulation. However, the proximal enhancers were extremely target specific, and they were not able to induce ectopic expression of corresponding surrounding genes. So our next question was, what causes this striking phenotype upon loss of the upstream boundary? And to answer this, we first performed region capture micro-C. And the first thing you see here is that this boundary here actually works as a loop anchor. So you can visualize these two loops, like the white arrowheads actually point to the upstream and the downstream loops. And you can see that these loops are absolutely lost in our mutant without the boundary. And there's domain fusion. And one more thing you see here is that there is this increase in putative contact with enhancers that lie in the corresponding boundary. And this is better visualized using promoter capture micro-C, where you can see that using FGF3 promoter as a bait, you can see that there is loss of interaction exactly at the boundary. And then there's increase in this long range contact with one of the ANO1 intronic enhancers here. And after this, we actually looked at the expression of FGF3, 4, and 15 in the midbrain. And what you see here is in the wild type, FGF3 and 4 is actually not expressed, and they are repressed well by deposition of K27Me3. However, by loss of the boundary, you see a massive upregulation of all three FGF ligands, and that recapitulates the expression pattern of ANO1. And we think, or rather we have shown that the enhancer can come in contact with these FGF genes, and that's what leads to this overexpression, and that's what causes the phenotype. We wanted to prove that, and for that we made another rescue model. So the first thing you see here is deletion of this boundary causes this phenotype, but when we deleted even this bigger chunk of DNA, which actually encompasses the boundary plus the enhancer, the phenotype is rescued, which clearly proves that the enhancer was contacting the FGF genes, and that's what was causing the phenotype. Now, because this CTCF cluster comprises of multiple motifs, we wanted to look at the individual contribution of the motifs, and we did two more genetic lines where we actually first deleted the C1 motif, which is actually facing the ANO1 enhancer, and it was striking that deletion of this single motif was enough to recapitulate the phenotype that we observe here. In contrast, when we deleted the other three motifs that are actually facing the FGF genes, we do not see the phenotype. So deletion of one motif towards the enhancer was much more impactful compared to deletion of the other three motifs that faces the FGF genes. So to summarize, what we found is that some domain boundaries are extremely sensitive to perturbation, but this was not true in all contexts, and it of course depends on multiple other factors like location of proximal or distal enhancers, the strength of the individual CTCF motifs that define the boundary, and so on. And we found that not all FGF related functions were affected, so we clearly saw that deletion of all these three boundaries had no effect on implantation because the proximal enhancers of these genes were very target specific, and they were not able to induce expression of the neighboring genes even in absence of the boundary. And what we saw was deletion of a single motif within this boundary cluster actually led to contact of the neural enhancer with the FGF genes that led to its ectopic overexpression, and this followed the phenotype. And we also found that loss of single motif had a stronger impact compared to the loss of the other three motifs. So can we now predict which domain boundaries are more susceptible to perturbation? One way to look at it is to look at the conservation of the domain structure, and this is published from the Bernstein lab, which shows that in gastrointestinal stromal tumor samples, CTCF bindings are actually lost, and there is hyperactivation of FGF signaling. And not just in humans, this FGF locus is actually conserved in throughout vertebrates, and here you see that duplication of this region actually leads to neural tube defects in ridgeback dogs. So we now think that conserved domain boundaries may be more susceptible to perturbation. So with this, I would like to thank everyone at the Genome Structure Unit of GSRU, and especially Pedro for his immense guidance, Nina for helping me with the projects. I want to thank Ariel, who was a postbac, who worked with me from the beginning of the project. And I also want to thank all the core facilities of NIH, without which this work would not have been possible. So thank you.
Thank you. Questions?
Very nice talk, Shrita. I'm still wondering about the enhancer deletion versus the motif deletion. You know, the effects that you saw were very different. How do you explain this?
The enhancer deletion versus the motif. So when we so one of the NO1 enhancer was responsible for upregulating the FGF genes, right? So of course, when we deleted the enhancer, we saw that it was completely rescued, and we didn't see it. But for the motif deletion, like, I think we were also surprised that in spite of two other boundaries that lies beyond FGF3 and 4, just deletion of that single motif was able to upregulate the FGF expression. So we don't really have any experiments to support like what's exactly going on, but there can be, I think, multiple factors. But what we do gauge is that if you delete that single motif, which is part of the domain where NO1 actually lies, the enhancer is somehow loose, like in 3D space, and it can somehow come in contact with the FGF genes.
I see. Okay.
So it must have a partner CTCF in the other end, right?
Yes, in the other side of the top.
What happens if you mutate that one?
We did not do that, but I believe, what do you mean by mutate? Like,
no,
you
I mean
delete?
Like, yeah. I mean,
so if you keep this intact and delete the upstream, like upstream CTCF motifs, it should not affect the FGFs. But I have to check what other genes are there.
The TAD might get still destroyed.
The TAD will get disrupted for sure, but in terms of gene expression, we cannot predict like what will happen.
What determines the strength of the boundary? Is it just CTCF motifs, or are there other things binding?
Well, there are a lot of factors, transcription factor, motifs, because when we deleted the whole cluster, we are deleting a big chunk, like 25 kb. So there are a lot of like intergenic spaces in between. So we know from different studies, from a cohort of studies from other labs, that transcription factor motifs are important. Of course, CTCF is important. Like, there can be several other factors. But we did not check directly at the binding of transcription factors in the boundary itself.
I was wondering if you have looked at differential binding of different factors.
It is possible, yes.
Hi. Have you considered that your results are complicated by non-CTCF boundary elements like ESSRB or yin-yang 1?
Yeah, just like I was saying, like, but I'm not sure if those transcription factor motifs, if you're talking about this boundary.
So this locus doesn't have any of those sites, yeah?
Yeah, it's like, because it's a big region, it's possible those motifs are there, but we did not check that.
So you are saying that see what is important, right, to see the phenotype. Hi. So you are saying that the C1 is important to see the phenotype. So have you ever tried to to
invert it?
Yeah, to invert the sequence?
Yeah, I mean, we were trying that, but we don't have that data right away. Like, yeah, but I think C1. So when you use guide RNAs in zygotes, what happens is automatically you get either deletions or inversions. So the inversions were rare. So I don't know what happens to the phenotype, but I guess the phenotype might not be there because it's inverted and the boundary is still there. So now there will be like 4 CTCF motifs facing towards the FGF gene, but we don't know what happens.
Thank you for the wonderful talk. And let's just again clap and thank everybody else who gave talks today so far.
And let's thank Leelavati for chairing the session. And we'll break for lunch and we'll be back here at 2:30.